Dear colleagues, Marcus at Home welcomes Dr. Will Herrington and Dr. Kilian Donovan in Oxford, UK, and Dr. Alexander Grabner and Dr. Daniel Edmonston in Durham, North Carolina, with whom we would like to discuss whether or not we should still consider the phosphaturic hormone FGF-23 as a treatment target in patients with cardiorenal disease. Dear Kilian, Will, Alexander, and Daniel, it's a privilege to host 4 global leaders in nephrology this morning. Dr. Kilian Donovan is first author of a recent large-scale Mendelian randomization study on the cardiovascular impact of FGF-23, which he conducted at the Clinical Trial Service Unit and Epidemiological Studies Unit of the Nuffield Department of Population Health of the University of Oxford. The study has been published as a CJSON paper a few weeks ago, and it questions whether FGF-23, which has been widely claimed in the last 12 or 15 years to be a major driver of cardiac disease and an emerging treatment target for cardioprotective interventions, particularly in nephrological patients, is really pathophysiologically involved in heart disease at all. Dr. Will Herrington, who is corresponding author of the same paper, is associate professor and clinician scientist at the University of Oxford and practicing honorary consultant nephrologist at Oxford Kidney Unit. He jointly leads the Oxford Population Health Renal Studies Group in Oxford, and he's chief investigator of the EMPA-KIDNEY trial, arguably the most important kidney study in 2022. Alexander Grabner and Daniel Edmonston are assistant professors at Duke University Medical Center. Dr. Grabner's major focus of work is nephrology, particularly cardiac effects of FGF-23. And based upon his work, he is together with Christian Faul, the major proponent of a prominent role of FGF-23 in cardiotoxicity. Dr. Edmonston is a clinical researcher who studies the intersection of kidney and cardiovascular disease in patients with chronic kidney disease, and he's co-investigator of the HILO study, an ongoing outcome trial on the prognostic impact of stringent versus lenient phosphorus targets in hemodialysis patients. 4 global leaders necessitate a somewhat longer introduction, but now I switch the microphone to my co-moderator, Dr. Seemann in Vienna.
Yeah, thank you, Gunnar, and again, thank you to each one of you. It's a very prestigious meeting that we have. So the reason why we discussed this potential meeting and this real meeting now was this paper that Gunnar just mentioned. Is this paper, so to say, the nail in the coffin of the FGF-23 saga? And maybe we can have your views on that. So maybe can you give us a short background why you did exactly this Mendelian study and what you found out and what are your conclusions?
Perhaps I could just explain the rationale for why we were first interested in FGF-23 as a trialist unit. So in Oxford, we're a trials group, primarily the renal studies group. We ran the SHARP trial and then most recently the EMPA-KIDNEY trial. But we're always interested in trying to look for modifiable causes of chronic kidney disease progression or cardiovascular risk, and particularly heart failure, as we think that is an area which is understudied in the field of CKD. And we were particularly excited when we discovered the molecule FGF-23 and its reports associated with cardiovascular risk. And the first question we asked ourselves is, could this be a causal risk factor? It was a very attractive risk factor because it increased early in CKD, paralleling the early increased risk of cardiovascular disease with modest reductions in estimated glomerular filtration rate, increasing, much earlier than you see changes in, for example, hemoglobin. And so we also observed, although we're not experts in animal models, links between levels of FGF-23 and perhaps left ventricular hypertrophy. When we think about trials, they're 4-year, 5-year type projects, a major investment of resource, time, and opportunity. You only do a small number of large trials in your lifetime. And so we want to be utterly sure that it's the optimum question to ask ourselves. So because there are so many analytes which are disrupted by reduced kidney function, there's an enormous number of molecules you could focus your life's energy on to try and modify. And so what we wanted to do as epidemiologists was to, before we started to embark on large trials, is to assess for ourselves whether or not we thought this was a very good target, or just a good target, or in fact a target we wouldn't want to study. And so we actually embarked on 2 types of epidemiology studies. And I think they're a very nice example of how you could approach this if you are also a trialist and epidemiologist. We haven't got the final answer, that's for sure. But we have got 2 steps towards asking ourselves whether or not we want to motivate ourselves and a global community to do a very large trial. I don't want to spoil the rest of the story, so I'm going to hand over to Kilian with that introduction.
So hello everybody, thank you very much for the very generous introduction. I'm Kilian Donovan. I'm a renal fellow or registrar in Oxford, and I've been working with Will on various epidemiological studies in chronic kidney disease for a number of years now, but we've had a particular interest in FGF-23 as all of you have also. So by way of background to this, I'm not going to talk much about the biology of FGF-23 because there are at least 2 people in this conversation who are far more qualified to talk about that than I am. But the interest as an epidemiologist arises from the fact that this is a hormone whose level rises very, very early in the progression of chronic kidney disease. It is probably the first marker of the mineral bone disorder. And it has consistently, strongly been associated in epidemiological studies with cardiovascular diseases. There is, as I'm sure we'll hear about later, direct plausible data from animal models and from in vitro models that there's a direct toxic effect of FGF-23 on the cardiovascular system. And this led us to want to try and see if we could test, using epidemiological study designs, whether or not the association between FGF-23 and cardiovascular diseases was a causal association. So we started back in 2018 by looking at the available observational data at that time. So we conducted a meta-analysis of all of the available observational studies and randomized trials that had collected data on FGF-23 and cardiovascular disease for their participants. And this was the main figure from that paper. We stratified the estimates by the population of interest. So we included studies from general population cohorts, from cohorts of patients with chronic kidney disease not on dialysis, and from cohorts of patients on dialysis. And you can appreciate from this plot that there is a consistent moderate association between higher FGF-23 levels and cardiovascular disease. And the way that we performed this analysis was by looking at individuals in the top third of the FGF-23 distribution in each cohort versus those in the bottom third. However, when we drill into this data in a little bit more detail, we come across some slightly more interesting findings. So not only do FGF-23 disease rise very rapidly in CKD, but also the distribution of FGF-23 values broadens as CKD progresses. And if we look at general population cohorts without kidney disease, the difference in absolute values of FGF-23, of intact FGF-23, between the top and bottom third of the distribution is of the order of 30 picograms per mil, something of that order. And if we look at the dialysis population, the difference between the top and bottom third of the distributions in those cohorts is in the thousands of picograms per mil. So we have a 2 orders of magnitude difference in the exposure between these 2 groups, yet we see an effect size that is comparable. And that shape of association is not something that we see in other parts of human biology. Usually, if we have a physiological variable that is associated with an adverse clinical outcome, the association is log linear in shape, and this is not consistent with that. Furthermore, looking at the available observational data at that time, there were associations with many other clinical outcomes, not restricted to cardiovascular disease. So published associations in general population cohorts with acute kidney injury, fractures, or serious infections. taking this together, the fact that we didn't really see a convincing exposure-response relationship in the observational data, and the fact that we saw many non-specific associations of FGF-23 with other disease states, we came to the conclusion at that time that a likely explanation of the association between FGF-23 and these diseases was residual confounding. And by that we mean confounding by other unmeasured aspects of CKD. So usually when these observational studies adjust for CKD, they simply adjust for EGFR, which as nephrologists we know does not capture all of the various aspects of CKD, or residual confounding by other variables that haven't been measured. There's also always the possibility of reverse causation in observational epidemiology like this. Perhaps people who have the beginnings of cardiovascular disease already, for some reason, that in itself is causing higher levels of FGF-23. So when we were thinking about study designs that could eliminate some of these problems and try and get at the real answer as to whether or not there was a causal link, Mendelian randomization is the obvious candidate. So I'm sure most of us are familiar with the design, but essentially it relies on the fact that during meiosis, when alleles are being allocated to gametes, there is a random allocation of alleles due to crossover, and if these alleles are associated with higher levels of a particular exposure, so in this case FGF-23, then in effect you have gametes and then subsequent individuals who have been randomly allocated to a higher level of FGF-23. And because this randomization process is occurring before conception, it is not vulnerable to reverse causation. So an outcome that develops later in life cannot influence your allele allocation at the time of crossover. And similarly, it's much less vulnerable to confounding by exposures encountered later in life. So the first step in a Mendelian randomization analysis is identifying the genetic instrument that you're going to use as your instrumental variable. So to do this, we worked with the SCOLOP consortium, which is it's run by Anders Madderstig in Sweden. So this is a consortium of many different observational studies and randomized controlled trials from across the world, but mainly North America and Europe, where participants have had 19,000 different analytes measured on a proteomics platform, and they've all been genotyped. And these are predominantly general population cohorts of patients. So this is our Manhattan plot from that genome-wide association study. So these were the main loci that we identified. The loci I've included in black here are loci that were also identified in a big GWAS that was published in 2018 in JSON, and the two loci in red were not identified in that GWAS, which is quite interesting, and we might come back to talk about that a little bit later. But obviously the FGF-23 locus, that's not surprising that that's appeared there. CYP24A1, that's an enzyme involved in vitamin D metabolism, so again, not particularly surprising that that would be associated with FGF-23 levels. And the ABO locus is referring to the blood group antigens. And this is RGS14 is a protein involved in G protein coupled receptor signaling, which again has been associated several times with FGF-23 levels. So from these variants, we need to define a genetic instrument. So we chose to select 34 independent variants that were associated with FGF-23 levels at this significance level here. And that choice was based on balancing, developing a sufficiently powerful instrument versus not selecting variants that were simply associated with FGF-23 by chance, and not selecting pleiotropic variants. And then for each participant in our subsequent analyses, we generate a genetic score, an FGF-23 risk score for each individual participant on the basis of their genotype at these variants. Now, obviously, you can't, for Mendelian randomization, you can't just choose any variants. They need to fulfill certain criteria in order to be valid instrumental variables. And these are the three cardinal assumptions that people sometimes talk about in Mendelian randomization. So number one, your variants need to be associated with the exposure of interest. So ideally, we would want to validate our score in a different cohort. And your genetic variants should be the relationship between your genetic variants and your outcomes of interest should not be confounded by any other variables. Now, this is not a directly testable assumption, but it is something that you can do your best to demonstrate that that's not the case, and I'll show you later on what we did to do that. And also, your variants of interest should only interest your cardiovascular phenotypes through the exposure of interest through FGF-23. So first of all, in order to validate that our instrument is associated with FGF-23, we collaborated with a research group in Canada who had run a randomized controlled trial of diabetes treatments, but fortuitously had measured FGF-23 on all of their participants, and they were all genotyped. So when we applied our genetic score on the basis of those 34 variants to the participants in this study, it remained associated with FGF-23. So this was an external validation of our score. And then in order to verify that our score was not associated with other cardiovascular risk factors, i.e. to make sure there was not a pleiotropic genetic instrument, we took the UK Biobank 340,000 participants, divided them on the basis of their FGF-23 genetic score into fifths. And you can see that across fifths of the genetic score, there was no association with these known cardiovascular risk factors. So having established that our instrument is associated with FGF-23 and is probably not confounded by associations with other cardiovascular risk factors, we then move on to conduct our main statistical analyses. So we primarily use the UK Biobank for our analyses. So this is a cohort that I'm sure you're all familiar with, but it's a very large general population cohort in the UK. They've all been genotyped, and their clinical outcome data is automatically collected prospectively from the NHS National Hospital Episode Statistics. So anytime these patients present to hospital, once they've left, their discharge clinical coding is all automatically incorporated into the UK Biobank. And a subset of these patients are recalled at intervals for surveys, and some of our patients had cardiac and vascular imaging. So within this cohort, we estimated that our genetic risk score would have a power to detect an 8% risk increase in atherosclerotic events per standard deviation of FGF-23. So in the observational studies, the risk that is observed is of the order of 20 to 30%. So we anticipated that we would have power to detect an effect of that magnitude. We also looked in these other very large publicly available data sets to ensure that we were not missing smaller effect sizes that we were not powered to detect in the UK Biobank. So these are the main results from our analysis. So the figure on the left shows the results in the UK Biobank, and the estimates on the plot itself are the odds ratio of developing this outcome per standard deviation increase in genetically predicted FGF-23. And we can see across atherosclerotic and non-atherosclerotic outcomes, there is no convincing association there. And on the right-hand side, we have the data from these large cohorts, so almost 1 million patients or 1 million participants in cardiogram, and similarly large numbers across megastroke and Hermes. Now, with Mendelian randomization, there's always the possibility that you're seeing something because of the way that you have designed your instrument, and so it's important to do sensitivity analyses and try and develop an instrument in different ways and see if that affects your results. So specifically, when you're looking at an exposure that is an endogenous protein, it can be considered more valid to only use variants that are actually genetic variants that are located around the gene of interest as your genetic instrument. And that's because these variants are likely to be affecting protein levels directly through gene expression rather than through other pleiotropic pathways that might bias your results. So that was probably the most important sensitivity analysis. And then we also conducted a secondary analysis where we removed any variants that had ever been published to be associated with cardiovascular risk factors. So this is our results using just the cis variants. So the confidence intervals are significantly wider because we only had 2 variants that actually were at the FGF-23 locus. But regardless, it doesn't look like there's any convincing associations there. I'm not sure what to make of this association with heart failure in the Hermes cohort. Probably just random chance. And then this is when we designed the score in a slightly different way and just removed any variants that were potentially pleiotropic. And again, the results were null across the board. So our concluding message was that FGF-23 is unlikely to be a significant cause of cardiovascular disease. And the findings that we see in the conventional observational studies are likely to be due to residual confounding. There are certainly some caveats to our studies. So number one, we did not measure FGF-23 directly in UK Biobank, so it hasn't been measured in that cohort. And that limited our ability to actually validate our score in the cohort of interest. And secondly, the UK Biobank is a general population cohort. From what we've seen before, we know that FGF-23 levels in this cohort are substantially lower than those that we see in patients with CKD or hemodialysis patients. So it is conceivable that the effects of FGF-23 are qualitatively different at extremely high levels that we see in patients with chronic kidney disease. And that was not a testable hypothesis within our study design. So thank you for listening.
So thank you very much, Kilian, for this exciting background in the study itself, which looks very straightforward. And now I'm eager to hear what Professor Edmonston and Professor Grabner will respond to this data before we maybe discuss it all together.
Thanks for giving me the opportunity to summarize a little bit of FGF-23 research from the past 20 years. I want to highlight observations on the pathophysiological role of FGF-23 in cardiac remodeling and heart failure, what we know and also what we do not know so far. I want to start out with a brief background about FGF-23 and FGF receptor signaling to set everyone on the same page, and also to highlight why ongoing research has been challenging to date. So we know that FGF-23 belongs to a pretty heterogeneous group of fibroblast growth factors, and there are canonical FGFs that act paracrine, there are intracellular FGFs that only act within the cell, and there's a group of endocrine FGFs that per definition act as hormones, so they're produced by one cell, go into circulation and act on a target organ. This endocrine group of FGF consists of FGF-15, 19, and FGF-21 or FGF-23. FGF-23 mediates its effects via FGF receptors, and the human genome encodes 4 different FGF receptor isoforms. FGF receptor 1 to 3 also can form different splice variants, and there's pretty convincing data that FGF-23 can basically bind and activate all 4 isoforms. So as a reminder, what does FGF-23 actually do? So it's bone-produced hormone that is basically the master regulator of phosphate homeostasis. So in response to serum phosphate, FGF-23 via the circulation acts on the kidney. It induces phosphaturia by downregulating sodium phosphate cotransporters, and it reduces intestinal phosphate absorption. How does it do that? It downregulates calcitriol synthesis by decreasing 1-alpha-hydroxylase levels, and also by increasing 24-hydroxylase levels, and reduced calcitriol levels then reduce intestinal calcium phosphate absorption. So what is very critical here is there is overwhelming genetic evidence that indicates that FGF-23 requires a co-receptor to mediate its effects on phosphate homeostasis, and that co-receptor is called transmembranous clotho. And there are numerous studies that are all mentioned here that demonstrate that if clotho is not present in the kidney, FGF-23 cannot induce phosphaturia. So, and based on those genetic studies, the prevailing concept for a very long time was that FGF-23 can only activate FGF receptor in the presence of clotho, and since clotho expression is very limited to the kidney, the parathyroid glands, or the choroid plexus of the brain, all other target organs cannot respond to FGF-23. And I want to summarize some data that questions this concept and mention that FGF-23 can actually bind and activate FGF receptors in the absence of clotho. And there has been data generated almost 20 years ago by a group around Ken Wright and then by Musa Mohammadi that shows that FGF-23 binds FGF receptors, and this is so those are surface plasmon resonance and then graphs from the most recent publication from Christian Fowles group. And what they did is they immobilized FGF receptor 4 on a chip surface and used different concentrations of FGF-23, and you clearly see a dose-dependent response in the light curves, indicating that at higher concentrations, more FGF-23 can bind to the FGF receptor. We have shown, and others have confirmed, that FGF-23 can bind FGF receptor 4, and this binding to FGF receptor 4 induces PLC gamma calcineurin and fat signaling. And here is a schematic that basically demonstrates in the kidney FGF-23 binds alpha-clotho FGF receptor complexes, induces RAS-MAP kinase signaling, which transcriptionally regulates phosphate excretion. In cells that do not express clotho, high levels of FGF-23 can bind FGF receptor 4 independently of clotho, and this activates PLC gamma calcineurin and fat signaling. So the main question is, okay, good, what does this do if that pathway gets activated? That leads us to the second observation. There are now numerous publications that demonstrate that FGF-23 exerts direct effects on cardiomyocytes. And I want to summarize some of the in vitro studies that have been performed. So there's convincing data that FGF-23 induces hypertrophic growth of neonatal and adult mouse and red ventricular myocytes. FGF-23 also promotes rhythm alterations in cardiac myocytes. There are some in vitro studies that suggest that FGF-23 induces fibrosis via activation of cardiac fibroblasts. There are several papers now that indicate that FGF-23 impairs calcium handling and contractile dysfunction in adult cardiomyocytes. And to also show some unpublished results that I generated here at Duke in collaboration with Nina Bursik. He's a professor at Duke Brett School of Bioengineering, and they bioengineer cardio bundles. Those are three-dimensional structures made out of myocytes and fibroblasts. They can be grown in a three-dimensional matrix. So we cultured those bundles, treated them chronically with FGF-23, and then we just sectioned the bundles and measured myocyte cross-sectional area. And indeed, those myocytes undergo hypertrophy. And we also perform functional measurements. We can basically measure the contractile force of those bundles, and chronic stimulation with FGF-23 decreases contractile force, and the contractile dysfunction could be prevented if those bundles were co-treated with an isoform-specific FGF receptor 4 inhibitor. Next, so the question is, of course, I mean, in vitro experiments always only tell us a very small aspect about what actually happens in disease, and there are numerous publications now that demonstrate that FGF-23 and FGF receptor 4 directly affect the heart, and I want to focus on A, CKD, and B, cardiac remodeling, because those are the studies where the strongest data exist. And there are now numerous publications that show that recombinant FGF-23 injections induce left ventricular hypertrophy in mice. So people have injected FGF-23 IV, people have performed direct intracardiac injections of FGF-23, and one study led by Professor Darrell Crowells, they injected FGF-23 IP, and in all those studies, the heart undergoes left ventricular hypertrophy. We have shown that mice that express an FGF receptor 4 gain-of-function mutation develop left ventricular hypertrophy. In contrast to those short-term injections, this mouse model takes a lot of time, and they develop LVH. They do not develop massive systolic dysfunction, but the LVH is there. Next, mice that are subjected to a high-phosphate diet develop high levels of FGF-23 with normal kidney function. And if we chronically feed mice a high-phosphate diet, they develop LVH. And mice with global deletion of FGF receptor 4, they do not develop LVH, so they are protected. Similarly, again, a group by Darrell Crowles, they show that mice with conditional deletion of FGF receptor 4 from the heart do not develop LVH in response to FGF-23 injection. And as proof of concept, there are basically 3 studies that demonstrate that pharmacologic inhibition of FGF receptor 4 or a pan-FGF receptor blockade or inhibition with calcineurin prevents left ventricular hypertrophy in CKD rats. And the important thing here is those pharmacologic interventions do not affect kidney function or blood pressure, so it's independent from the kidney effect, and the intervention also do not lower serum FGF-23 levels. And finally, to sum this up, a small gap in the literature. It's incremental research, but it's unpublished data from my lab. So we fed mice with a global deletion and also mice with cardiomyocyte-specific deletion of FGF receptor 4 and adenine diet for 16 weeks, which is known to induce LVH. And in contrast to wild-type mice, global FGF receptor 4 knockout mice do not develop LVH, as indicated by LV-mass here measured by echoes. And similarly, in mice with myocyte-specific deletion of R4, they also do not develop LVH compared to wild-type mice. And of note, kidney function and FGF-23 levels are unchanged in those mice. So this leads us to the next observation. Do all animal models with FGF-23 excess develop cardiac remodeling? And the clear answer is no. There are now several publications that show that very likely outside of CKD, FGF-23 and FGF receptor 4 do not have a prominent role in the development of cardiac remodeling, and be happy to discuss the reasons for that later. So it's basically Renal and from Vienna, they deleted FGF-23 and clotho from mice and performed pressure overload experiments and on a rescue background, and basically there was no difference in mice that have or do not have FGF-23. So pressure overload induces heart failure and cardiac remodeling, and the deletion of FGF-23 had no effect on it. Then second, a pretty controversial topic, we know that patients with X-linked hypophosphatemic rickets develop hypophosphatemia due to primary FGF-23 excess, and the clinical data is also not entirely clear, and so is the mouse data. So some models show, some papers show that those mice develop cardiac remodeling, others do not, and it's unclear what is the reason for that, and definitely further research is needed. So next, a group from Hanover, they generated a transgenic mouse that overexpresses FGF-23 directly in cardiac myocytes, and at least in healthy mice, they were not able to see any form of cardiac phenotype, so there is no LVH, there is no contractile dysfunction in those mice. And finally, which fits in the narrative, it's already a couple of years ago, we here at Duke performed myocardial infarction experiments, and our hypothesis was based on a manuscript by Renal Erben's group that showed that myocardial infarction in mice upregulates circulating FGF-23 levels. So of course it would be intriguing to speculate that the circulating elevations in FGF-23 also contribute to cardiac remodeling or heart failure in myocardial infarction. And what we did is we deleted FGF receptor 4 from mice globally, induced MI, and I think the results are very clear. The mice develop heart failure, the mice undergo dilatation, cardiac dilatation, they develop LVH, but there's virtually no difference in wild-type and FGF receptor 4 knockout mice. So this leads us to a summary and the outlook. So what do we know? I think there is, from a basic science perspective, there's pretty convincing data that in FGF-23, elevated serum levels of FGF-23 bind and activate cardiac FGF receptor 4 in the absence of clotho, and this activates PLC gamma calcineurin NFAT signaling, which contributes to cardiac hypertrophy and potentially cardiac remodeling and heart failure. So the main question is, of course, what do we not know? And can time and dose-dependent responses explain the differences in animal models with and without CKD. There are CKD-specific mechanisms that modulate the FGF-23 effects on the heart. And if we think about it, is it vitamin D deficiency, hyperphosphatemia, hyperparathyroidism? Soluble clotho plays a prominent role in CKD, and we know that clotho deficiency directly contributes to a variety of pathologic outcomes in CKD. And recent data indicates that soluble clotho and FGF-23 share mechanistic pathways. So the question is, can soluble clotho deficiency directly modulate those pro-apoptotic effects of FGF-23? And then another big question is, we know that FGF-23 undergoes proteolytic cleavage. And the question is, of course, how and why is FGF-23 production and cleavage regulated in cardiomyocytes? And ultimately, is there actually a physiologic role for FGF-23 effects on the heart, or do we only see those so-called off-target effects in patients with CKD when FGF-23 levels are substantially elevated? And with this, I'll hand over to Daniel.
Well, first I want to just, again, echo my thanks for the invitations. It's wonderful to talk about this topic, and it was actually extremely wonderful to hear, Kilian, your overview of yours and Professor Herrington's work. It's pretty outstanding. So I'm just going to provide a few thoughts or observations, if you will, on observational data for FGF-23 in cardiovascular disease. And I think to start, it's important to understand that, building on the data that Alex presented, that the associations between FGF-23 and cardiovascular outcomes tends to align pretty well with what we understand from the mechanistic data, and that is the strength of the associations between FGF-23 and left ventricular hypertrophy or left ventricular mass and FGF-23 and heart failure tend to be of a stronger association than those demonstrated for things such as vascular calcification or atherosclerotic cardiovascular disease, myocardial infarction, that sort of thing. Not to say that there's no data to support some associations there, but that the data usually is more convincing that the associations are stronger for these other outcomes. Just looking at a little bit of the data in support of this, some of the data for the association between FGF-23 and left ventricular mass index and left ventricular hypertrophy comes from Christian Fall and Miles Wolf, their group back in 2011, which first demonstrated this in the chronic renal insufficiency cohort, or the CRIC cohort. It's been replicated in other cohorts, both with CKD and without CKD, with an important caveat that I'll mention again here shortly. The association with heart failure was also demonstrated. The independent associated heart failure was also demonstrated in the CRIC cohort and studied by Julia Ciala and Miles Wolf group back in 2014. But this has been replicated in other CKD cohorts and community cohorts, such as the MESA cohort and the ERIC cohort, atherosclerosis risk in communities cohorts as well. So to kind of summarize a little bit for observation one here, the observational data does seem to align pretty well with mechanistic data, and to extend that further, I'll pay off the caveat that I introduced just a moment ago, and that's cohorts that include both participants with chronic kidney disease and without chronic kidney disease tend to demonstrate a stronger association between FGF-23 and left ventricular mass, or FGF-23 and heart failure, a stronger association of those with chronic kidney disease than those without, either shown formally through statistical methods such as interaction terms, or at least inferred through stronger differences in effect sizes for the CKD versus the non-CKD group. Here I'm showing some data from the cardiovascular health study, which is community-based cohort of older adults, but it's been replicated in other cohorts for left ventricular mass index and left ventricular hypertrophy. And Joe X's group has shown the same association difference in CKD versus non-CKD for the cardiovascular health study as it pertains to heart failure. So extending this a little bit further and kind of tying back to what Alex was talking about, the preclinical models also support a stronger link between FGF-23 and cardiac pathology in states of secondary FGF-23 excess, specifically chronic kidney disease. And the observational data does tend to support that as well. So Dr. Donovan gave a pretty outstanding overview of Mendelian randomization in general, and definitely their excellent work in this area. I'm not going to reiterate any of that, but I did want to briefly touch on a couple of the other Mendelian randomization studies in the FGF-23 space to really highlight some observations, but also introduce perhaps a couple of caveats as they more or less specifically pertain to FGF-23. So first, findings from one Mendelian randomization study to another can really differ pretty widely. And I think Kilian had hinted to some of this, and that it really depends on the genetic variants chosen for your study and the instrument used and validated, and specifically how much of the variants in circulating FGF-23 levels are explained by those variants or collection of variants. And that can widely influence statistical power and the inferences that you can actually make from Mendelian randomization. That's an important point, I think, in general, but definitely with some of these studies for FGF-23. And I'll present a couple of other more general thoughts on Mendelian randomization in a couple of slides. But another point to make here is that Mendelian randomization findings can really diverge substantially from preclinical data and often have findings that don't quite yet at least have a plausible biological mechanism. So one example here, the study by Henry and colleagues published in Circulation that showed a strong association between circulating FGF-23 level and higher odds or higher risk, I should say, of heart failure in their observational component of their study. But when they analyzed the genetic variants linked to FGF-23 level, those variants were associated with a lower risk, actually, a substantially lower risk of heart failure, an inference that would suggest that FGF-23 is protective with heart failure, and it's not something that's really been substantiated, at least in a mechanistic sense of preclinical models. Also, this study by Lang and colleagues highlights some other considerations. First, again, showing here that for coronary artery disease, that there is a bit of a lower risk of an association with a lower risk of coronary artery disease with genetic variants linked to FGF-23, which again really does not have much basis in preclinical or mechanistic data to support that. And here, the inferences, the associations with heart failure seem to vary a bit based off of the statistical method chosen to evaluate the instrument, with a couple of the methods demonstrating definitely a very null association, but with one method showing perhaps a trend towards, but not statistically significant, observation of a link with higher risk of heart failure. And then this study, also Kilian had briefly mentioned, this Mendelian randomization study by Aquo and colleagues published on CJASN was interesting for the use of a polygenic risk score for low EGFR. They used this to stratify participants, and after doing so, they were able to demonstrate a strong relationship between variants linked to FGF-23 and heart failure for those in the low EGFR strata. Which was actually kind of interesting. And in concert with some of this preclinical and other observational data, I would say this finding does support perhaps a unique relationship between FGF-23 and, in this case, heart failure for people or cases of chronic kidney disease. And additionally, with this phenomenon being demonstrated, at least in a roundabout way, using this Mendelian randomization approach, it would suggest that perhaps this relationship persists beyond the concerns of residual confounding, at least as it pertains to this relationship. And then the last thoughts on Mendelian randomization in this space, to achieve the level of evidence that from a Mendelian randomization study that's sufficient to make causal inferences, you definitely have to satisfy some assumptions that Kilian brought up before. And I wanted to highlight where a couple of these might have a sticking point for FGF-23. The first assumption is that the discovery and validation of your variants is going to be reliant on demonstrating a strong and stable association with, in this particular case, a circulating FGF-23 level. And most of the Mendelian randomization studies have used the intact FGF-23 assay, which I think is important to at least acknowledge because there are some caveats that come along with that. First, a lot of the observational studies demonstrating very strong associations between FGF-23 and cardiovascular events have done so, applying the C-terminal assay rather than the intact FGF-23 assay, although there's definitely data linking intact assay levels with cardiovascular outcomes. But with the intact assay, there's some data to support there's perhaps more intra-individual variation, diurnal variation, or perhaps some greater influences by delays in specimen processing and that sort of thing. Additionally, there are some Mendelian randomization studies that have developed or validated their genetic risk scores or variants in cohorts that include participants with both or either an intact or a C-terminal FGF-23 assay. And there's not really a lot of great data on, or at least information in the paper about how these different assays were handled, whether or not they were excluded or conflated, which would definitely play a role in diminishing whether or not the assumption 1 was met. Kilian definitely touched on a lot of the concerns for assumption 2 about the concomitant risk factors and how that's a very difficult thing to definitively demonstrate. And to satisfy this requirement, you often have to know the risk factors to look for and have a robust method to adjust for them. And again, the ability to overcome that assumption really is not, or at least demonstrate that you've overcome that assumption is not really any better methods to do so than are applied in traditional observational studies. So that's always a bit of a caveat there. Another caveat is, it's also been mentioned, these are often conducted in large population-based cohorts. And so when it comes to looking at cardiovascular outcomes specifically, in this case, heart failure, that often leads to using a claims-based definition of heart failure, which may not perform as well as a clinically adjudicated definition of heart failure that you would see in something like the CRIT cohort. And then lastly, as has been mentioned a few times now, the focus on a general population setting means that Mendelian randomization is not going to capture whether if there's a threshold effect for FGF-23's influence on cardiovascular events, here heart failure. And the AQUO study definitely would suggest perhaps in favor of this hypothesis. And given this possibility, I think we can't really extrapolate a lack of a causal relationship between FGF-23 and heart failure beyond the general population. The door is still open that there may be a specific and unique relationship, even causal relationship in the CKD population. So that's just summarized here in that observation. And then lastly, I just want to touch on a couple of slides here about treatment. And so I think there's data that direct antagonism of FGF-23 through something like anti-FGF-23 antibodies is definitely not a viable option to study in humans in states of secondary FGF-23 axis like CKD, since we know animal models that have this sort of antagonism have premature calcification, mortality, and other secondary sequelae from derangements in mineral metabolism that are just too dangerous. But there is some data from therapies that secondarily lower FGF-23 that don't influence kidney function, but are still associated with improvements in either left ventricular hypertrophy or heart failure. So one piece of data in this direction is from a secondary analysis of the EVOLVE trial of cinacalcet versus placebo in people with receiving maintenance hemodialysis. The participants who received cinacalcet had more of a decrease in FGF-23 than those without. And when you focus on those with greater than 30% decrease in FGF-23 levels in response to cinacalcet, there's a lower risk of cardiovascular events, especially one of which I should say is heart failure. And then the last example I'm going to touch on here is in a randomized trial here of alpha calcidol, which is a vitamin D analog, versus etelcalcetide, which is somewhat of a calcimimetic. This demonstrated less left ventricular hypertrophy progression for the group randomized to the vitamin D agonist versus etelcalcetide. And where this is important is that these agents share overlapping influences on PTH, but have a divergent influence on FGF-23, whereby the etelcalcetide will lower FGF-23 or tend to, and the vitamin D analog may increase. And so since there's no changes in kidney function, overlapping influence on PTH, perhaps this diverging influence on FGF-23 is what's resulting in this difference in LVH progression. So the last observation here would be therapies that indirectly lower FGF-23 levels. These examples pertain specifically to end-stage kidney disease, may suggest improvements in LVH or heart failure with that strategy. And with that, I will say thank you again for the opportunity.
Thank you very much, Professor Edmonston.
Well, maybe we give the Oxford group some minutes to reply. And kind of trying to bring all this together.
Maybe I could make a general comment, which it's as I think that all research has limitations. I'd say the key limitations of the Mendelian randomization experiment that we did with FGF-23 is the fact that we studied FGF-23 concentrations in a general population, and so we can't really talk about the dialysis population. It remains entirely possible that the extreme levels of FGF-23 that you observe in dialysis populations could still be causally associated with left ventricular hypertrophy and heart failure. And so we haven't been able to address that. It would have been much more attractive to start to think about addressing it if we observed some form of association between FGF-23 in the general populations, because we see with the classical conventional epidemiology, rather robustly consistent associations with heart failure in those populations. But we also, as Kilian highlighted, see non-specific associations. And one of the things we harbored for a long time is the concept of residual confounding. Residual confounding probably by unmeasured or undermeasured or imprecisely measured kidney function. So you couldn't adjust for levels of kidney disease, or indeed simply regression dilution bias, under measurement of the natural variation of blood pressure, so that any adjustments within the models that are done in conventional analyses under adjust. And I was really struck by the Henry circulation figure that was shown, where there was essentially a comparison of conventional associations on the same forest plot as Mendelian randomization associations. And it was alarmingly common for them not to line up, and in fact there to be moderate to large associations in the conventional approach, with Mendelian randomization associations being much more modest, and in one case qualitatively in the opposite direction. And that to me it reinforces a long-term suspicion that what we really need to do in modern day epidemiology now is to go from the animal models, and when you have robust, repeatable evidence that a biomarker might be causally related in certain animals, and it may be applicable to humans, and then we have the epidemiology, which suggests in conventional analyses that it is strongly associated, that we very rapidly move on to assess whether or not we can test the association in a Mendelian randomization experiment. And I think where we have major limitations at the moment in our fields is being able to derive suitably powerful instrumental variables within which to be able to test these hypotheses. I think what we need is large data sets, having these biomarkers that we're interested in measured, genotype data sets, so that we can derive through GWAS, polygenic risk scores, to then apply in large data sets and assess causality before we move on. This isn't always going to be possible. Where it is possible, we just need the resource. So it'd be fantastic if one day UK Biobank had FGF-23 measured. We had to go to the Scallop Consortium to try and generate the worldwide evidence on FGF-23 and its genetic architecture, and to be honest, we could have had a stronger risk score. And so I think we need to think about that as as funding agencies and as scientists, to push for that type of phenotyping, so that we can rapidly assess newly identified biomarkers in a large-scale way and in a randomized way. I've always been a believer of large-scale randomized trials. We need to do even larger randomized experiments before those trials now, because it could be incredibly efficient. And I wanted to just sell the idea of using these data sets. UK Biobank's open access. It has got prospective follow-up for a lot of vascular outcomes. Heart failure aside, we've done a lot of validation work looking at atherosclerotic cardiovascular outcomes, comparing them to adjudicated data sets, the data in the and we get, excellent, agreement. So these data sets really are incredibly valuable. heart failure is an area which does need much more attention, and is particular attention in our field. And I think if people can start to validate that outcome in UK Biobank to make sure it really does measure the different types of heart failure we're interested in, then in the coming years we'll be in an excellent position to be able to test rapidly biomarkers associated with the CKD. So that's my sort of my vision for how we can move from basic science and animal models through epidemiology to identifying the best targets for collaborative large-scale trials.
Alexander, Daniel, are you aware of anybody who's planning a clinical trial, at least in dialysis patients, on FGF receptors in the next years?
So
Definitely not for the antibody, no.
Because I mean, the antibodies are there, they are used in oncology. So if we still believe in this, this would be an opportunity. And maybe from my part, yeah.
I mean, I had, when I moved to Duke, I mean, our paper just was published and I was really excited. And also, I mean, Duke has a strong interest in commercialization, and it's just very challenging to like move forward. And the main problem that at least I encountered is that, I mean, we've published the mechanisms, we have a lot of the models, but in the end, we don't have a real intellectual property, right? We don't have an IP. An IP would be the compound, and the compounds have been basically generated by others. And it's also, there is, I think in the cancer field, the excitement for FGF receptor inhibitors is like faded down a little bit. I think they've not been as convincing as the people initially thought. And I think also like those startups, they just don't really have an interest in like entering a field that ultimately, even if we show strong preclinical data, and even if a phase 1 clinical trial looks good, I mean, what will if the compound moves forward, we need a phase 3 clinical outcomes trial, and that is just like money only a handful of companies in the world can provide. And I think that it's a high risk, high reward project, and also with SGLT2 inhibitors, basically just like turning the nephrology and cardiovascular field upside down. I think there's just no real interest. And then of course, well, I think we need to take like 3 steps back when it comes to outcomes trials in nephrology, and probably Daniel can tell you more about this. I mean, we need to first see what is the role of phosphate. If we lower phosphate, how does it affect FGF-23? I mean, we've come up with a pretty cool pathophysiologic mechanism, but ultimately also, even if we could show now that even if those Mendelian randomization trials would be positive, I mean, we currently do not even really have like the tools to lower FGF-23, right? What to effectively block it? I mean, we just don't have it. I mean, phosphate binders do not really do it. Phosphate restriction does not really work. So it's like we are still, there is still like so much research needs to happen before we can like make a define comment on this. And it's, I mean, I'm excited to be part of this, and I definitely want to push forward this, because also from the basic science perspective, our understanding how FGF-23 targets the heart, what does it do? It's, I mean, we've seen associations that could suggest actually beneficial effects of FGF-23. Is there any evidence in any form of preclinical studies that this might be the case? And I hope that some papers will be published in the upcoming years to maybe or maybe not show that. So there's definitely a lot going on.
Just to briefly discuss, you had mentioned FGFR for antibodies and that sort of thing. I mean, I think if they're going to have a role for study, it would probably be in the pre-dialysis CKD rather than in dialysis. And I say that because the first line of therapy for these people receiving maintenance hemodialysis is to target the derangements of minimal metabolism. And the first step we need in that direction is to actually validate that those approaches have that lead to positive changes in clinical outcomes. And the HILO study is one, and there are hopefully others down the road that are trying to demonstrate that as well. But I think that's going to be the primary focus in that population. So in the population that such as the pre-dialysis CKD, we know that targeting minimal metabolism without derangements in that population probably is not going to be the way to go. And so, approaches such as the, maybe the FGFR receptor antibodies would be the most that would be the most viable population for study of that. But again, all the reasons Alex just mentioned, there's going to be some trouble getting those types of studies off the ground.
Kilian, are you planning to do other Mendelian randomization studies in the field of CKD, MBD? So looking at phosphorus or vitamin D?
So I've no immediate plans to do any further studies in the mineral bone disorder field. No, but we have other bigger plans.
Kilian, so we're very interested in Mendelian randomization and what it can offer. There are some statistical challenges to doing it, but we're really interested in trying to work out, as trialists wanted to do kidney disease progression trials, on identifying what we say causal markers of kidney disease progression. And Kilian's spending the next 3 years doing that using various data sets.
So my last question, Daniel, when will we see results from HILO? Because again, we are really looking forward to learn about the effects, if any, of phosphorus binders.
Yeah, we're still actively enrolling. Obviously, there were some shifts from COVID and other influences, but HILO is still actively enrolling. So we don't have a specific date on that, but we'll keep you updated.
All right. So we're looking forward to it again. Thanks a lot. This was a fantastic academic hour. And thanks so much, I guess, from all our colleagues who can really benefit from your points of view. And we're looking forward definitely to more from Oxford, more from Duke in the upcoming years. Thanks so much.
Thank you very much.
Thank you.
Thank you so much. Nice to see everyone.